Right, this is going to be our second snow day lecture of the semester. Unfortunate because we aren't meeting in person and giving a chance to ask questions. So, first thing is it's going to be important to write down questions that you have from this and for new class. In this situation, you don't have to send your notes to me. This class meeting or this recording is as important as any of our other class meetings, and you should take notes and write down questions and so forth, but you don't need to send me the notes because everybody's missing class. And so this is what we're at. You should start reading chapter 17. I think I'll get really close to starting to talk about chapter 17 by the end of class today. However, we've been talking about it since day one. And our first exam is actually now officially 2 weeks from today. I think it's Thursday, the 9th of March. So that's where we're at. And that's what's the exam will cover everything up to what's on this list here. And where we're at right now is we just started talking about heat shock transcription factor. So it's another transcription factor, and we want to reduce it and explain how it's regulated, and then in turn how it regulates the rate limiting step of the heat shock genes. The rate limiting step of the heat shock genes is they have arrested RNA polymerase. And with these other examples as well, there are hundreds of transcription factors that help assemble the pre-initiation complex. MyoD is one of them. There are hundreds of transcription factors that remodel chromatin. Estrogen receptor is one of them. There are hundreds of transcription factors that release unarrested polymerase. Heat shock transcription factor is one of them. What these all have in common is that in each case, these transcription factors regulate the rate of transcription by regulating the rate limiting step. And in the genes that they regulate, the rate limiting step is that. Then these transcription factors regulate the rate limiting step in their target genes, and in their target genes, the rate limiting step is that, and so on and so forth. Okay, so that's where we're at on that. Then I just want to show you this. I wrote this up new. I think of chapter 9 as having 3 pieces for us. The first is we already know a lot about this chapter because of the clay model videos, and so obviously they're going to be a big part of our exam. We're becoming experts on transcription, regulation, and transcription factors. Then in this chapter, what we're in the middle of diving into is this, and we've talked about pre-initiation complex, MyoD, we talked about remodeled chromatin and estrogen receptor and other nuclear receptors, and we're just starting to talk about this one. And then the last piece for us that I'll talk about today in class here is that there are a series of methods that the book uses, and I add a couple more methods to that list to talk about some of the many ways that scientists study the regulatory region of genes and the transcription vectors that bind to them. All right, and so again, this is our first piece, right, from this chapter, and then the next piece is because MyoD fits into there, right? I talked about this. Here's MyoD, and TBP binds to MyoD, and then MyoD helps get TBP to where it needs to be. TBP is one of these transcription vectors that bind to the promoter of the myogenin gene. Okay, and so there's that pre-initiation complex, and TBP is one of those proteins. These are all different proteins. I don't know why they have the same color here. And so that's that. And then when we turned to the estrogen receptor, we were doing a few things at the same time. We were introducing another transcription factor that's regulated by ligand, and that's how it's switched on and off, as are the other nuclear receptors. And then we were also introducing estrogen receptor itself, and the protein, and how we learned about tamoxifen. And tamoxifen is an antagonist of estrogen. And so we learned about the specific sum about estrogen receptor, and then we learned about the rate limiting step in the response genes are regulated by the estrogen receptor and estrogen, and they are regulated by remodeling chromatin, and so we know something about that. And so I said, we could just switch these labels around, and I could have made our starting clay model video about the estrogen receptor and estrogen response genes and how they get activated. Okay. That's really important to appreciate. And so here we are in chapter 17. So like I said, you should start reading it because we are going to be super experts on chapter 17. And so we use the estrogen receptor to learn some about histones and nucleosomes. And this is a nucleosome. The DNA is wrapped around it to help condense it, to help shorten it, so it can fit inside the nucleus of a tiny cell. And so that's where we're at. And we've learned a lot. And we went into a lot of detail about these pieces. Okay. So, I added a couple more slides, I guess. So, tamoxifen is one, then a structure of the nuclear receptors, then, introduce this idea of these very important nuclear receptors in human health. Then we have this evolutionary tree of nuclear receptors and how they're related to each other because they are paralogs. So we have that very important term that we define. So lots and lots of stuff that we've been learning about. Then in the context of remodeling chromatin, we need to learn more in the book about remodeling chromatin. And to be able to tackle it, what I said is we'll focus on 3 topics in chromatin remodeling. One is DNA methylation. We've talked about that already a fair amount in our course because DNA methylation happens during differentiation. So that's one of the early topics we covered in the clay model videos. Then this is a new one to us, histone acetylation, and these changes in chromatin structure, these terms and stories. And then in the context of a heat shock transcription factor, we're going to get into this last example, and this is a transcription factor we'll talk about, GAGA factor. Okay, so let's get into the heat shock transcription factor. And here's the story with the heat shock transcription factor. The heat shock transcription factor is a transcription factor, and it is like MyoD in that it has a DNA binding domain. And this DNA binding domain binds to a heat shock response element. A heat shock response element, so just a name for it, you could call that an enhancer, and it's part of the regulatory region for heat shock genes. We talked at the end of class last week is that there's going to be a signal when there's a lot of protein denaturation in a cell because of some kind of stress, heat, or some other kind of chemical stress. There's going to be a lot of protein denaturation. And the product of the heat shock genes are going to help refold those, help renature those, and help rescue the cell from otherwise going to die very soon. So here's the heat shock response element that the heat shock transcription factor is binding to. Here's the DNA binding domain. And here's just the first concept for this one, is that there it is, it's binding. But in this case, the heat shock transcription factor has a low affinity. It doesn't bind very well. It doesn't stick very well. It kind of is like a hand that grabs hold of weakly, and is not strong, and it just really frequently lets go. So we have a low affinity DNA binding domain that binds to the HSC, the heat shock response element, upstream of heat shock genes, okay? Which you could also call the enhancer of these genes. And then it's going to have a transacting domain. And that transacting domain's function is to release the arrested or paused RNA polymerase. And scientists discovered this 30 years ago. And they still don't know, to the best of my knowledge, they really don't know how this gets that done. And so we're not going to go into the details of this, because there's some data about it, but it involves kind of describing other pieces of the transcription story, and we're just not going to go into it because it's not really well characterized. But definitely, low affinity DNA binding domain, then this transacting domain, okay? And then you'll see that there's just another region here that will be important, and you'll see why in a second, and it's called the trimerization, and we're going to call it a domain. It's just a very short region, okay? And so here's what happens, and we'll just draw this a little bigger, and that is that here is the, in an unstressed cell, heat shock transcription factor protein is always present. So in every one of our cells, and in every animal cell, in every plant cell on our planet, there's always this heat shock transcription factor, always present. And that's because it needs to be ready to respond to this heat stress or this stress, really rapidly, okay? And this is what it looks like, and this is kind of a fanciful drawing, of course, but here's that trimerization domain, and the protein is folded like this, so that the trimerization domain is covered up. And so the protein is floating around, and it could bind to the DNA, but it doesn't bind with very high affinity. So most of the heat shock transcription factor is just in the cytoplasm. It's not even in the nucleus. And there's a couple reasons why that's true, and we'll learn a second one later in our course. The first one is it just has a low affinity for the enhancer, the heat shock response element, the HSE in the nucleus, those enhancers on the heat shock genes, and it just doesn't bind there strongly, so it's mostly just out in the cytoplasm, and it's folded this way, like that. Okay. Now we have some kind of stress happen, and proteins get denatured, and this protein is a really stable protein. It is very hard to denature it, but when we have this stress for denaturation, the heat shock transcription factor actually changes shape. It doesn't destroy it, it just kind of unfolds it. And so what happens is we get a protein shaped like this now, and now this trimerization domain is exposed. And what happens is we actually get, it's hard for me to draw this, but I'll draw it like this. We actually get 3 heat shock proteins. That's why it's called a trimerization domain. It's going to be a protein trimer. So we get a protein trimer. And so this is non-covalent bonds, just a stickiness between these molecules. That wasn't happening here because the domain was covered. And there's this conformational change during this stress. And the HSF molecules join together in these heat shock trimers. Okay? So now I'm just going to redraw this and show you that they continue to have this low affinity for the DNA. But when we go back and look at an HSE, HSEs always come together in groups of 3. And as a result, this heat shock trimer can bind there with 3 binding domains, and looks like that. So that's what HSF looks like when it binds to the heat shock gene. And now it's still like a low affinity binding for each of these. But this has become, and I use this analogy, it's kind of weird, but it's like a 3-handed rock mountain climber where, it's pretty hard to climb up, a cliff or rocks. But if you had 3 hands, it would be much less likely to let go and fall down, right? Because every once in a while you let go with this hand or that hand or that hand, but when this one lets go, there's really typically 2 others holding on. So we go from individual low affinity DNA binding domains to this trimer that is a high affinity. And these HSCs are aligned like this, so they can all bind together. And so that's what happens to activate this, is we get trimerization. And the effect of that trimerization is we create a molecule with surprisingly high affinity. So now it's going to get, when it gets in the nucleus, it's just going to really stick on these heat shock response elements and do a great job. Okay? So keep that in mind that here we have the heat shock response elements, and they're there all the time. And but the individual HSF monomers have low affinity, and they just don't stick very long when they bind. This is much higher affinity than 3 times higher affinity, because it goes back to this idea that that when one of these lets go for a moment, the other two are here, still holding on, and this one is held in place so that it will probably likely bind again really soon, not rather than float away, right? Okay, so that's the whole key for that, and that's the central story of the heat shock response, is that the heat shock transcription factor functions like a thermostat, like a little stress switch to detect stress and protein changes in protein structure. Again, the heat shock transcription factor is a pretty stable protein, so when other proteins in the cell get completely denatured, completely unfolded, what happens to the heat shock transcription factor is it changes its shape to expose the trimerization domain. We get these protein trimers forming that have effectively a high affinity, like that. So I have just a couple of slides. Here's that HSF just floating around. This image down here just shows that it's mostly in the cytoplasm. We've got it tagged with a green tag, and they just can't stay in the nucleus very effectively. But then this stress causes change of the shape, and that's that red trimerization domain that's covered here. Gets uncovered, these guys trimerize. It's a detail that they also get phosphorylated. And that's a second story that we're not going to learn about today. And we bind to this response element that's a series of 3 heat shock transcription factor binding sites. And we really rapidly start to accumulate the HSF in the nucleus. And it's in the nucleus now because it binds to the DNA in there and stays in the nucleus. Here's just another image of that, showing a little bit more, and just showing that what's happening with these phosphorylation is just this is a way to turn off the response. So it's a feedback loop where we're going to turn it on and get active, and then to turn it off, we're going to remove these phosphate groups. And shut down the response. HSP70 is actually a heat shock gene. It's one of the products of the heat shock response. So we have a nice sort of activation and then repression, turning off this response once the heat shock proteins build up. And that's all there is to that. But we get to add it to our list. I guess I don't have the numbering on here slightly correct, but heat shock transcription factor is a really beautiful example of the dynamic story of transcription regulation and regulated based on these transcription factors. And that's it. And so then again, in this case, we don't learn the details of how the transacting domains of that heat shock trimer release this arrested polymerase. But they do. And the heat shock response is a very rapid response, a very strong response. So as soon as this RNA polymerase gets going, another one takes its place. And when this arrested polymerase gets going, there's already another one landing and forming that pre-initiation complex, and it moves forward, and it doesn't even really slow down because the heat shock transcription factor just keeps it going past that arrested polymerase point. Now, okay? So then the last piece of this is that we just want to talk about this third thing. So we got DNA methylation, we got histone acetylation, and then this nucleosome phasing example. And let me just go back to an image of these nucleosomes. So our book shows nucleosomes. And the nucleosomes are these histones, collectively are called a nucleosome, with the DNA wrapped around them. Okay, like that. And so when let's go out here and draw a picture of this idea. And these so here's a set of these nucleosomes. And then here's the double-stranded DNA. So double-stranded DNA, like that, and it just wraps around them like that. And then it wraps around this one like that, and wraps around this one like that, wraps around this one like that. And these are the core histones that make up the ball, and then there's an additional histone here called the linker histone. And what happens is that this actually helps package the DNA up even further. So we get these kind of pulled together in a group like that, and they can be very inaccessible in the form of heterochromatin, or they can be more unfolded a little bit more like this in the form of euchromatin. Okay, so you can see this, and then let's just do this and show you this, that for the heat shock genes and out of any other gene, the gene is folded into a somewhat shortened version because of these nucleosomes, and in the case of the heat shock genes, up here we would have, let's just draw a little bit more, and we would have this being the heat shock response elements, where the heat shock protein is going to bind. And if we look in the unstressed cells, like all the cells in our body right now, we would see that upstream of these heat shock genes, the heat shock response elements, they don't have heat shock protein bound because it's low affinity, but kind of curiously, they all have like the DNA is in this really available organization, so that the moment that we trimerize, there's space there for the heat shock transcription factor to bind. And that's because there's another transcription factor. So I'll just draw a little bit more DNA like this. And then it goes into some more nucleosomes. So near to where the HSEs are, there's another response element, another enhancer region here. And that tends to have a sequence that includes GAGA, right? So that's just, the ATCG sequence. And if we look at the detail of these sequences, this tends to have a GAG sequence, and it's an enhancer. Helps regulate the gene like that. Transcription factors bind to it. In this case, the transcription factor that binds to it has a good name. So it's called GAGA factor because it binds to that GAGA sequence. And when we looked in the unstressed cells, what we see again is, well, the HSF is not here because it's low affinity monomers in the cytoplasm. But in unstressed cells, the GAGA factor is already here. And the role of the GAGA factor is to make sure that there are no nucleosomes right here. So there's always, every second of our life, a nice binding site for these heat shock transcription factors if they ever get activated. Because remember, again, there's a lot of pressure on to rescue the cell. When the cell gets stressed, we have these heat shock transcription factors get activated by trimerization, and they need to bind now. And so the role of GAGA factor is to do that. And so we get these nucleosomes, and in every cell, the nucleosomes on a heat shock gene are in the same position. And they're said to be phased. So they're in phase. So see, we have them like this. And in every cell on the heat shock genes, the nucleosomes are always in the same place, the same phase. And that's because GAGA factor sets up the nucleosome binding. And so that's the role of the GAGA factor. And so GAGA factor is cool because it makes sense, and it's it's important. It's always in our cells, and that's what it's doing. And it also shows us a different kind of transcription factor, right? Because GAGA factor has no transacting domain. It doesn't regulate transcription directly. It helps get these nucleosomes positioned to keep the DNA of the heat shock response element, the HSE, available for immediate binding. And so it's another story about chromatin, right? And in our usual stories about chromatin, we talk about chromatin and chromatin remodeling. We want to change these folding of these nucleosomes by adding acetyl groups or removing acetyl groups or changing other modifications, right? And in this case, that's not what's happening. In this case, we're just getting the nucleosome set up permanently in a position that allows the heat shock transcription factor to quickly add. So GAGA factor is a really cute transcription factor, and we add it to this. Okay, so our main reason for talking about heat shock transcription factor is, what's an example, our favorite example of a gene transcription factor that regulates genes by helping release arrested polymerase. Yay. So that's why we're talking about it at this point. But then heat shock transcription factor is a fascinating transcription factor because of how it gets activated. So we add it to our list. And so let's just go find that list. So we add it to our list. It's a different transcription factor. How are these dynamic? Well, it gets switched on by how it changes its folding. Great, good one. And then we're not going to add GAGA factor to this list because this is a story of the dynamic switches. And the GAGA factor doesn't switch. The GAGA factor is there, and it's helping always to keep these nucleosomes in the proper position. So it's a good one to show that we like this list because they're really good examples of dynamic stories about how transcription factors get switched on to regulate gene expression. Maybe by phosphorylation. Maybe they're allowed into the nucleus. Maybe they just simply get made. Maybe they have a ligand. Well, with GAGA, none of that happens. It's just always bound and always helping prepare for transcription. And it's really important in the heat shock response, even though it doesn't switch on and off. Okay, so there it is. Great. And that's what we're going to talk about that. And so let's see if we can get back to this. So that we're done talking about this set. And so let's get to where we're at. There's the heat shock transcription factor, then that. And so we're back to this. When we talk about remodeling chromatin, we emphasize these three stories, and that's what we're going to talk about nucleophasing. There are lots of other examples of nucleosome phasing. GAGA factor is a great one for us. Just remember that, and it's a good one for the reasons I just described. It's got a nice name, too. Okay, so here's this third piece I want to talk about that's dealt with some in this chapter, and it's examples of how transcription factors and the regulatory region enhancers are studied. And there are a lot of different ways to study them, but we're going to talk about 5 methods, and they're mostly covered in Chapter 9, but we'll talk about them in just, in a series like this, 5 different methods. And so the first one is, let's say, do I have this yet? Oh, sorry, I somehow had this slide out of alignment. But here it just shows the GAGA factor it's just a different abbreviation of it, but that's GAGA factor. See, and it's already there, and we already have RNA polymerase there arrested like that, and so it's a paused RNA polymerase, and we get stress. So we're going from this situation to this situation. This is unstressed cells, every one of our cells in our body, and then if there's a stress, get some protein denaturation. That causes the HSF to partly change its shape. We expose them to trimerization domains. They trimerize, and they're looking for the HSE. And look, GAGA factor has these nucleosomes out of the way, so it can bind there quickly, release the rate-limiting step, and get that transcription going of these heat shock genes. Okay, so I just had that out of time. So here's these DNA deletion studies, and just to, well, we're just going to say, we're not going to go into this, but it's just a different way that sometimes people use the word promoter, and I guess I just won't try to introduce that here. Just stick with, we're going to look at the regulatory region. And the way we're going to do these is we're going to use the estrogen receptor as our transcription factor. And the estrogen receptor binds to estrogen response genes, and sometimes I call them target genes because that's just a loose term. And I thought I should probably talk about a specific estrogen response gene. And so we're going to talk about one. There are a bunch. Okay, there are a bunch, and so this is where the estrogen receptor is going to bind to the estrogen response element, or the enhancer, upstream of this gene. And we're just going to learn about the name of one of them. But remember, there are many response genes, right? Our bodies really change as we go through puberty, and that's because a lot of different gene expression occurs. Okay, so this is one, and it's one that's not even in us. The gene is called vitellogenin, and it's a really good example. And vitellogenin is a protein, and the protein is referred to as yolk protein. And so humans don't have yolk, so this would be protein that's found in non-mammals, animals that are not mammals, that have eggs with yolk. Like what? Like a chicken. There's a lot of vitellogenin in chicken, in the yellow part of the chicken egg. There's a lot of vitellogenin. And vitellogenin is called a yolk protein because it gets put in, and it's a nutrient. And what it is like is it gets formed as a long protein, and in the embryo, it's going to get broken down to amino acids to provide amino acids for the embryo to build the proteins it wants to build. So yolk protein, or vitellogenin, is really a way to store amino acids for the embryo to use. And who makes it? Mom. And where does she make it? Not in the egg, it turns out. So some animals will make it in their liver or other cells, and it will be carried through the blood to the egg, and it will be brought into the egg and stored. And they make it elsewhere because there's a lot of it. And here's the great thing about vitellogenin is the cell makes a lot of it. So vitellogenin gene, wherever it is expressed, has a really strong regulatory region that is regulated by the estrogen receptor. And so when there is estrogen, animals like birds make lots of vitellogenin. So that's just one example that we use to talk about this, okay? All right, so years ago, let's just see where we're at. So years ago, we'll just use this as a drawing of this example. And so this could be the vitellogenin gene. Vitellogenin gene. So then somewhere up here, there must be an estrogen response element. And this is a famous gene that scientists use to start to study the estrogen receptor, because when they added estrogen to these animals, they got lots of vitellogenin protein, yolk protein made. So this must be an estrogen response gene. And it's a good one to study because it responds really strongly. So where is the estrogen response element? And the answer is we somehow have to identify it. And the way that scientists would do that is they get a piece of the gene DNA in a test tube and they would start to randomly remove parts of it. So I'm just going to draw down here that the first piece they get in a test tube is like this long. And that's the first deletion they do. And then they delete a little bit more. And that's the second one they test. They delete a little bit more. That's the third one. Delete a little bit more. Fourth one, and so forth. And then they take these DNAs in a test tube. They put that first one into a cell. And then they add estrogen, and they check to see if they get the vitellogenin RNA made. And then they do that experiment for the second one with a different set of cells. And when scientists are doing this, instead of trying to put the DNA into an animal and make a transgenic animal, they just have some cells in a dish because they just want to do a quick experiment to see if this form of the gene is responsive to estrogen. And so what they're doing is they're putting these deletions into cells and testing them. And they just put them in there briefly, so scientists call this a transient expression assay. Transient just means brief or temporary. And our book talks about this some. And it's just involved to try to study these. And what you're doing here is you're mapping the location of the estrogen response element because this first one has it, and so the estrogen receptor can bind here and activate the gene. And the second deletion has it, so that can activate it. And the third one still has it, but just barely, and it can activate it. And the fourth one doesn't have it. So this is called a deletion study because we're just deleting portions of the gene. And the goal of a deletion study is to identify an enhancer. So we didn't know that. And so years ago, scientists took the vitellogenin gene from chickens, they did a deletion study, and they found which piece of the regulatory region was active. And these were active, and this was inactive, as were the subsequent ones, the even shorter ones. And so they were able to say, well, the estrogen response element is right here. So that's a really sensible way to identify the location of the enhancer. So that's our first experiment that we want to learn about is a deletion study. And that's what deletion studies are like, and that's what they're done. So in an exam, I could say, give an example of a DNA deletion study and what's it used for. And you could say, it's used to map the enhancer in the regulatory region of a gene. And the way it's done is to just randomly delete portions of the gene and test them to see if they still have that activity that the enhancer would provide. And if they do, they must have the enhancer. And if they don't, they must not have the enhancer. It allows us to map the position of enhancers and map other kinds of regulatory regions also. Okay? So that's our first method. And again, it's to map the enhancers for a gene. And the example that if we were going to describe, if it says, describe deletion studies and give an example, say, well, you could use that to map the estrogen response elements upstream of a vitellogenin gene. All right, so next one, nuclease footprinting. And this is has very similar purpose. It is to help map the enhancer. This one uses a different strategy. This one is like saying, let's just take some DNA in a test tube, and if we add the transcription factor, it should bind. And it should bind to the enhancer. Nucleases are enzymes that break down DNA. So the nuclease will destroy the DNA. And what happens is nuclease would protect this region. A transcription factor would protect this region. And that's why it's called a footprint, is because in this case the estrogen receptor on the vitellogenin gene will protect from degradation this portion. And so to study it, what scientists do is they compare the experiment where they add nuclease to just DNA with no transcription factor versus DNA with the transcription factor present. And the way they detect these this degradation is they just let go for a short time so that we just get some random pieces of DNA. And what scientists do is they will add to the experiment, they'll add a radioactive tag at the end. So they'll be able to detect the pieces that get made. And there's a figure I have to just show you what the result is like. And that is that if we just take the these are two different figures showing the same thing. And so if we just take DNA and we start out a long piece of DNA and we add a little tag to the end of it, in this case they added some radioactivity, and then we randomly cut the DNA, we would end up with pieces of DNA that were longer and shorter and shorter and shorter. And we could load those fragments onto a gel and separate them based on size. So this is gel electrophoresis, just separating fragments based on their size. And the shorter molecules move faster, and the longer molecules move slower. So we'd see radioactivity like this along the length of it. And this is the sample where there was no transcription factor. And so we see each of the pieces represented. And then when we do it with the transcription factor present, the transcription factor would bind on a specific location and protect that region from degradation. And so we would see a kind of gap where there are no fragments of this length because the transcription factor estrogen receptor, in our case, bound to them and protected the DNA from getting cut by the nuclease degradation enzymes. And so we would see this, and people call that a footprint. So that's a second way that scientists could map the location of the enhancer. First one is this DNA deletion work. Second, totally different kind of experiment. Try to protect the DNA at the enhancer by adding the transcription factor and protect it from degradation. And I'll describe one more method, and that's this gel mobility shift assay, and that does the same type of thing. And so scientists want to be really convinced of the location of an enhancer, so they use different methods to come to the same conclusion. Here's what this is like, is we could take the vitellogenin gene and we could, in a test tube, just cut it into pieces like this, and then we could add our transcription factor and it would bind to just one of the pieces. Now what we could do is do this experiment and either add the transcription factor or leave it out. So I have two samples like this, And what we do is run these on a gel. So gel electrophoresis would separate these fragments based on their size. And so we'd see them running like this. And in the case of the gel mobility shift assay, what we'd see is if we added the transcription factor, what that does is it makes this DNA molecule larger, it has a greater mass, it's going to move more slowly. And so what we'd see is with this, when we ran this result on a gel, we'd see 3 of the 4 fragments, but then this fragment would appear to be missing. We'd see it up here running more slowly. And it's because it has a transcription factor bound to it. So that's why it's called a gel mobility shift assay. So the mobility was shifted. And what this does is it again maps the location of the enhancer. The enhancer must be this fragment because we added the transcription factor, estrogen receptor, and this is the fragment that was affected. And I have a slide of that too. It's from our chapter, and here it just shows one fragment instead of several fragments, just to keep it simple. And so it says here's the DNA by itself, and the DNA fragment is moving this fast. And when the transcription factor is added, the mobility of that piece is going to slow down. In my example, I just used more pieces just to give the feeling of, okay, so there are several pieces. Which one of them is the important one? And what you'd see is there are 3 pieces that still run the same speed through the gel, same mobility, but one has been slowed down. So that's that, and that's 3 different ways of helping to provide evidence for where the enhancer is. And the purpose of that is that like a molecular biologist doesn't really want to think about an enhancer like we did in the clay model videos. Instead, they want to think about the data that supports the understanding that the estrogen receptor binds to a specific part of the vitellogenin gene. So I'm going to stop here, and on Tuesday we'll pick these up, and then we'll get into chapter 17 because it's the last part of chapter 19 we want to talk about. So 2 more methods in chapter 9, and then answering any questions you have. So again, make sure you write them down. And again, you don't have to send your notes in for this, but please, take notes every time you do anything. Okay, great. And so that's where we're at for now.